Thank you so much, and thank you everyone for having me, and thank you to the organizers of the Targeted Protein Degradation webinar series for the opportunity to speak to you today about the clinical data for ARV-110 and ARV-471, which are orally bioavailable ProTACs currently in clinical trials for the treatment of prostate and breast cancer, respectively. As the focus of this research forum is protein degradation, I feel a debt of gratitude to all of you on behalf of the patients who are already showing signs of benefit from all of your hard work. It's a privilege to share these data with you today. And in a time of global tragedy, I hope this will provide a little ray of sunshine to energize you all and to remind you of how impactful your work is to people suffering with life-threatening illness. What I won't be sharing, though, as you just heard, is the chemical structure of our ProTACs, because luckily for me, that was just disclosed this past weekend at AACR by Larry Snyder, the head of medicinal chemistry at Arvinas, and he joins me here today. I have to digress for a quick moment before I jump into the exciting stuff and add that on a personal note, it's great to be back home with the Dana-Farber folks. I spent almost a decade on Binney Street, including several years on the 6th floor of Mayer, in the lab of Alan DeAndrea, which I don't think is there anymore. And I worked on polyubiquitination rather than monoubiquitination in the Fanconi pathway. As a minor little nugget for those of you who live in the Boston area, one of the perks of working in the Longwood area is that if you're lucky enough to be pregnant and happen to go into labor at work, it's literally like a hop, skip, and a jump across the street to one of the two greatest hospitals in the US or beyond, particularly for labor and delivery, although there's a fierce rivalry. Anyway, I happen to know that from personal experience, so if you want to use that in recruitment materials, you'd be surprised. It could help. In any case, I'm currently an employee and a shareholder of Arvinas. Here are our safe harbor and forward-looking statements. I don't need to spend a lot of time on this. I think you all know how it works. At Arvinas, we use our ProTAC platform to tackle indications in oncology, immuno-oncology, and neuroscience with two ProTACs, namely ARV-110 and ARV-471, currently in clinical trials. Behind our clinical assets, we have a broad and deep portfolio of programs in various stages, some of which are shown on this slide. I'd like to start at the beginning, which for the clinical chapter of heterobifunctional protein degraders starts with ARV-110. An orally bioavailable androgen receptor degrader ProTAC, which is being studied in the clinic for the treatment of men with metastatic castration-resistant prostate cancer who have progressed on standard approved therapies. ARV-110 is a potent and selective degrader of ARV in VCAP and other cell lines that are typically used to interrogate androgen receptor binders and degraders. The half maximal degradation concentration of DC50 or DC50 for ARV-110 and VCAP cells is 1 nanomolar. Not only does ARV-110 potently decrease the abundance of the androgen receptor via the ubiquitin proteasome system, it does so with good selectivity, as demonstrated by the proteomics data shown. ARV-110 also demonstrates efficacy in vivo, and really in vivo, which I get to tell you about a little later. In an enzalutamide-insensitive PDX model, ARV-110, when dosed once daily at 10 milligrams per kilo PO over the course of a 20-day study, demonstrated 100% tumor growth inhibition, which compared quite favorably to enzalutamide dosed at 20 milligrams per kilo once daily. In addition, you can see on the right, plasma PSA levels following ARV-110 treatment significantly decreased versus mice treated with either vehicle or enzalutamide. The favorable in vitro and in vivo efficacy data, coupled with the appropriate preclinical risk mitigation assays and a lot of CMC work, resulted in the FDA allowing us to take ARV-110 into the clinical setting. This is an overview of the current clinical treatment paradigm in the US for prostate cancer, which is actually rapidly evolving. Patients with prostate cancer often progress through multiple stages of the disease, starting with castrate-sensitive, either localized or metastatic disease, and eventually reaching metastatic castrate-resistant. Some portion, a large portion, who have localized disease up front, I should say, are cured, but those who are not go on through these stages. Androgen deprivation therapy has a long history of use in prostate cancer, and therapies like chemotherapy, docetaxel, or cabazitaxel are used predominantly in mCRPC. Second-generation therapies such as enzalutamide are also used in mCRPC, but have gained approval in earlier lines of therapy in recent years. Unfortunately, none of these therapies are curative, and so eventually patients run out of options for disease control, leaving a high unmet need in metastatic castrate-resistant prostate cancer, where patients need non-chemotherapeutic options and novel therapeutic approaches. It's important to remember that our phase 1 trial of ARV-110 is in very late line mCRPC patients, later really than any of the other trials on AR therapies that we've seen. So what does that mean? Well, enzalutamide and other AR-directed therapies rapidly decline in efficacy when used in series. Basically, you develop mechanisms of resistance. And so we know that if you give enzalutamide after abiraterone or abiraterone after enzalutamide, you don't get nearly as much benefit. They start with a robust, perhaps 90% reduction of PSA, the biomarker prostate-specific antigen, as much as 80% response rate in the castrate-sensitive setting. But by the time the patients progress on to a third-line metastatic castrate-resistant setting, the PSA 50, which is the decline of the PSA level to 50%, has dropped to single digits, really 8 to 15%. In the late lines in which ARV-110 is starting, nobody exactly knows what the efficacy should be, but it's expected to be around 10% or lower. Excuse me. Our overall strategy is to develop ARV-110 in earlier lines of mCRPC, and that we'll be exploring as well. But I'll tell you a little bit more about that in our phase 2 later today. Our phase 1 study of ARV-110, really what you all want to hear about, was designed with a standard 3+3 dose escalation at a starting dose of 35 milligrams daily. Inclusion criteria include men with mCRPC, at least 2 prior therapies, and evidence of disease progression on the most recent therapy. The primary endpoint is safety and defining a maximum tolerated dose, as well as a recommended dose for the phase 2 studies. We also looked at pharmacokinetics, anti-tumor activity, and some exploratory biomarkers. These next few slides I'm going to share with you are based on the first 22 patients that we treated on study and were presented originally last May at ASCO. The baseline characteristics for these 22 patients are shown here. The main thing to note is that the patients are all heavily pretreated, as I mentioned earlier. All the patients received AR-directed therapy with abiraterone, and more than 3/4 also received enzalutamide or other. There are 2 other agents in this space as well. Additionally, 3/4 also had chemotherapy, and that was comprised mostly of docetaxel and cabazitaxel. On average, patients received a mean of 5 prior regimens. So how did the safety do? The treatment-related AEs are presented in this slide by dose and broken down by grade 1, 2 versus grade 3 or 4. In general, ARV-110 is very well tolerated, with the great majority of AEs being grade 1 in severity. GI-related toxicities such as nausea and diarrhea were most common, with approximately 25% of patients reporting these symptoms across all dose levels. However, they were usually quite mild and did not lead to treatment discontinuation. Notably, the grade 3, 4 AEs were infrequent and limited to 3 patients, one of whom experienced a DLT at 280 milligrams and an AST and ALT rise of grade 4. Additionally, there was a second patient at a dose of 140 who had to de-escalate and re-escalate also with LFT abnormalities. And in a really interesting moment of clinical development, it was really an astute, our CMO, Ron Peck, who was just coming on at the time, recognized that Crestor was being used concomitantly with ARV-110 and not in any of the other patients. And they really were able to figure it out with further in vitro studies that ARV-110 inhibits a transporter, BCRP, which is critical to the uptake of rosuvastatin and quite a few other drugs. And so now we're able to manage that drug-drug interaction risk prospectively. So what else did we learn from these patients? And that's highlighted there, the 2 patients with the LFTs. Well, here I can show you really the first evidence of PROTAC protein degradation in a human subject. This is a specimen stained for androgen receptor before and after treatment with 280 milligrams daily of ARV-110. And as you can see here, there is much less dark brown on the right after 6 weeks of treatment as compared to baseline. When we looked at a little further at how long patients stayed on ARV-110, you can see here, this is again from ASCO, you can see here that patients really were already showing that they could tolerate the drug and were staying on it for quite a while. The table on the left here is really just to remind you that these patients had seen a lot of therapies. But a picture, as always, says it even better. And we were really amazed to see a patient who had really quite significant tumor regression after 6 weeks of treatment with ARV-110. Actually, excuse me, after he stayed on it for a very long time, but we saw the effect very early on. On the left, you can see that the patient has significant adenopathy, as shown here. In fact, so much so that this is the inferior vena cava and it's being compressed. After his therapy, 4 cycles, you can see that there's significant reduction in the adenopathy. So all this white junk basically is gone. And the IVC is now nice and round and patent. When you can see on the left, this patient had really received quite a few therapies, including chemotherapy, enzalutamide, abiraterone, and really had significant decline in the biomarker PSA. So these data got us very excited thinking about our phase 2 study. And with promising activity and a tolerable AE profile, we really honed in on our pharmacokinetic exposure and our molecular data to help design a trial, a phase 2 trial that would help inform the way to derive optimal benefits for the patients and their specific tumors, rather than a one-size-fits-all approach. In the next few slides, I'll share with you some of the data that informed our phase 2 design. So the exposure data I mentioned are shown here. The 2 orange lines represent preclinical efficacious thresholds. The lower one is the standard VCAP model, and the higher one is a VCAP model that is enzalutamide resistance. As shown on the right side of the graph, the 420 milligram dose was the first dose where we saw the plasma exposures consistently above the minimum level associated with preclinical efficacy. And 420 also exceeds the AUC associated with efficacy in the enzalutamide resistant xenograft model, which we've always thought would be an important target, since this is essentially the patient population in the phase 1 trial. So we were really happy to be able to consistently achieve these higher exposures at 420, and that led us to choose 420 as our recommended phase 2 dose. We also believe that achieving this threshold is important because we observed an exposure activity relationship. So in this slide, we're presenting the best PSA change, which is the standard way that in prostate cancer trials, people follow either tumor changes or the prostate-specific antigen biomarker. And to orient you, the graph goes from increasing exposures from left to right. So over here we have folks who had a minimal, these were below that lower orange line I just showed you. These folks were in between the 2 orange lines, so between the regular VCAP model and the minimum threshold of the enzalutamide resistant model. And then these folks were above the minimal efficacious threshold for the enzalutamide resistant animal model. And what you can see is that on the right, we see, so we'll start at the left. You can see that those patients really didn't do that well and had very few or little PSA reduction. When we move one panel over into the middle again, we can see significant PSA reductions, including 2 patients who had responded and were disclosed during our ASCO presentation. What I'm showing you here is actually more data since ASCO. On the right, when you what you can see is that the exposures that are higher really seem to show more evidence of a PSA response. We have 2 more patients who have a PSA 50 response, and we also have several that have a PSA 30% response. So there's a really, what I think cool, third cool dimension to ARV-110's activity profile. And we're really seeing it in this late line population. And that has to do with tumor heterogeneity. As the patients progress and the tumor becomes more resistant, you see genetically heterogeneous with really an increasing number of AR-independent mutations. As we've noted, the patients in our phase 1 trial have had many prior therapies, and we're seeing signs that this genetic context could be an important determinant of response. As a reminder, our phase 1 profiling showed that 84% of patients had at least one non-AR gene mutation. And as you'll see, many had far more than that, up to 6 in a panel of just 70 genes analyzed. So it's likely that many of these tumors are AR independent, with a lower likelihood, therefore, of showing a PSA response. All of that is to say, we really shouldn't have expected to see almost any response with with an AR degrader. To look at this a little bit more, you can see here that we're showing the best PSA change for all the patients with exposures above the minimum preclinical efficacy threshold. So that was the people in the prior slide from the 2nd and 3rd groups, along with their AR mutation status, including ARV7, a splice variant, and the number of non-AR genes altered. All the way to the right, you can see that this group includes the 4 PSA 50 responses that were seen on the prior slide. As you can see in the bottom row of the table, the vast majority of these patients have at least one non-AR gene mutation, and some had multiple distinct mutations. So again, really confirming what we knew, that this is a heterogeneous set of tumors. Despite the heterogeneity, we did see PSA declines in most of these patients, and that's really encouraging. The PSA 50 rate here is 14%, which is above the single-digit response rates that we would have anticipated. In the next 2 slides, we're going to focus on 2 populations in particular: patients with wild-type AR expressing tumors, highlighted here. Those had T878 and or 875 AR mutations, and that together makes up about 71% of the patients in this exposure-defined group. At ASCO, we showed that the 2 PSA 50 responses were in patients with tumors that had both T878 and H875 AR mutations. Since then, we've enrolled 3 additional patients, and with that mutation profile, and we've seen a few interesting observations. So all these patients have T878 and H875. First, the graph shows 4 of 5 patients had PSA reductions, including 2 deep PSA responses, one of which, as I showed you, had a RECIST response. That's the tumor reduction. Second, as you can see in the top row of the table, each have multiple AR mutations present, at least one of which is 878 or 875. In the middle of the graph, you see 2 patients that had PSA declines over 30%, despite having multiple mutated non-AR genes present, as well as the L702H AR mutation, which I didn't mention in this talk, but is one of the AR mutations which ARV-110 does not bind to and degrade. One of these patients also expresses the ARV7 splice variant, which is also not degraded by ARV-110, but that has a whole distinct biology to itself that we can presuppose we might see activity in. Lastly, as you can see at the bottom of the table, 3 of the patients, these 3, including one of them who only had a PSA 30, were on treatment for over 6 months. And this is a median for this subset, which is very encouraging, since the expected duration in this late line setting is between 3 and 4 months, based on recently published studies. Looking at the bullet points on the right, the important takeaway is that the presence of multiple AR mutations could be a signature that these tumors retain a significant level of AR dependence, which is perhaps surprising, such that therapeutically relevant levels of PSA reduction and durable time on treatment can be achieved even in the context of high tumor heterogeneity and in a setting where other AR inhibitors or modifiers such as abiraterone or enzalutamide have already failed. So patients with tumors containing T878 and or H875 mutations are being enriched for in the phase 2. And we believe that this could be the basis of potentially an accelerated approval pathway, which is really exciting as it may mean both a meaningful therapy for patients and getting it to them faster, which is really what this is all about. Now we're going to turn to look here at the patients with tumors expressing wild type AR, which is in most cases, the majority of patients. And they also have non-AR mutations present as shown in this table. Importantly, there are 2 PSA50 responses in this heterogeneous population as well, both at the 420 milligram dose level for a PSA response rate here of 13%, which again is above the single-digit response rates that we've highlighted earlier. The key point is that these responses demonstrate that ARV-110 has a much broader activity profile than just the mutation-bearing population, and it really also supports going into potentially earlier lines of therapy where the tumor may not yet have accumulated so many non-AR dependent mutations. These phase 1 data have pointed us toward a phase 2 study that will look both for the molecularly defined patients who may benefit, as well as the broader MCRPC population where we've also seen activity. We really couldn't have asked for a more exciting signal in a phase 1. I mean, just for what it's worth, the people in the investment world really want to see, huge response, but the investigators, our clinicians out there in the universe treating patients are unbelievably excited that in this late line we can see activity. In designing the ARDENT phase 2 expansion, we built in several features that are described in the top box. First, we want to establish efficacy in the populations where we've seen signals in the phase 1, especially in the 878, 875 patients, and perhaps broader, actually, AR mutation signature. We also want to continue to explore earlier and more AR-dependent populations. We're not excluding any patients in the design of this study. It's really still exploratory to understand who benefits from what, but they're all being placed in 4 subgroups shown on the lower left so that we can really define them later and analyze them. In summary, really more exciting things to come. I'm going to turn now to our second child. That's my pediatric background coming through there. We don't play favorites. This is ARV-471. ARV-471 is a potent degrader of ER in multiple relevant cell lines, including MCF47, with a half maximal degradation concentration, or DC50, of 1.8 nanomolars. And it's worth saying that we really benefited from learnings from 110, ARV-110, and we're able to be more efficient in the selection of ARV-471. Additionally, ARV-471 exhibits superior tumor growth inhibition. You can see that here, versus fulvestrant, which is a standard of care. And this is in a tyrosine 537 to serine patient-derived xenograft model. When ARV-471 is delivered orally on a daily basis to mice previously implanted with these PDX tumors, you see a nice dose-dependent tumor volume reduction superior to that which is observed with fulvestrant alone. And to connect the dots, in a corresponding Western blot analysis of the tumors post-takedown, which is not shown here, we saw a 79% and 88% reduction in estrogen receptor in the 10 and 30 milligrams per kilo arms of ARV-471, compared to only 63% reduction in the fulvestrant arm. And then we took it one step further and combined with palbociclib, which is a potent CDK4/6 inhibitor, and the effect is even more dramatic. You can see here in the combo line at the bottom that we saw 131% tumor growth inhibition, which is better than either palbo alone or fulvestrant plus palbociclib. So going a little bit back to breast cancer as a disease entity. As some of you may know, it's the second most prevalent cancer in women. 1 in every 8 women will be diagnosed with breast cancer at some point in their lifetime. Each year, over 200,000 women in the US are treated for breast cancer, and about 1% of new breast cancers are also diagnosed in men. 80% of breast cancers express the estrogen receptor. Interestingly, or not so interestingly, I got a call last night from one of my dearest friends from high school telling me that she just got pathology back on her breast cancer and wanted to discuss how to navigate. So it's really everywhere. The foundational therapy for ER-positive breast cancer are treatments collectively known as endocrine therapies that directly or indirectly target estrogen receptor. While existing endocrine therapies are reasonably effective, they do have important limitations. Despite this, no new endocrine therapies have been approved since the ER degrader fulvestrant was approved nearly 20 years ago. Recently, the combination of targeted therapies combined with existing endocrine therapies have become commonplace in the management of advanced ER-positive breast cancer. This is particularly notable for CDK4/6 inhibitors, which are really now established as the upfront standard of care for initial treatment of advanced breast cancer. Despite these advances, breast cancer remains a major public health issue due to the shortcomings of current therapies that we believe can be addressed by more effective targeting of the estrogen receptor. Unlike fulvestrant, ARV-471 is an oral therapy and is a much more potent and effective degrader of ER than fulvestrant. We believe that with this profile, ARV-471 has the opportunity to become the future standard of care in ER-positive breast cancer, or at least we hope, because if it really is as good as we're hoping and as strong as we saw in those animal models, it'll really be very effective. Before taking you through the data on ARV-471, I'll briefly describe the design of the first-in-human trial, which is similar to what you saw for ARV-110. We employed a traditional 3+3 dose escalation design, and the primary and secondary endpoints are typical for an oncology phase 1 study, focusing on safety, a recommended phase 2 dose, and pharmacokinetic and biomarker data. The inclusion criteria for the trial are shown here. A very important aspect of the eligibility criteria in this trial was the requirement that all patients have received prior CDK4/6 inhibitor therapy. And that's really unlike trials that have recently been reported in the selective estrogen receptor degradation class of investigational agents, known as SERDs. This criterion was actually recommended by the FDA, and it reflects the place that CDK4/6 inhibitors now have in the management of ER-positive breast cancer. The other requirements are listed on the left of the slide. And the critical point here is that the tumors that progress on CDK4/6 inhibitors display a high level of ER-independent resistance. Specifically, recent molecular profiling studies show that nearly 70% of patients treated with these agents develop mutations across multiple pathways that invariably lead to complete resistance to treatments targeting the estrogen receptor. This translates, of course, into extremely poor outcome for the patients in these situations. So how did our patients look? Well, here are the baseline characteristics of 21 patients treated with ARV-471. And this really, again, similar to what we saw with our prostate cancer patients, this really underscores the extensive amount of prior therapy that these patients received. And just to mention, these data were presented in December 2020. Most notably, patients had a median of 5 prior lines of therapy, and 100% of them, as I already mentioned, had already received CDK4/6 inhibitor. Additionally, over 70% of patients also received the ER degrader fulvestrant, and nearly 40% had received chemotherapy. So how did we do with safety so far? Well, I have to say ARV-471 has been remarkably well tolerated, almost so much so that we have been taken somewhat aback, because when you work in oncology, it's just, part of the game that nothing's for free, and you're not going to, you're not going to get anywhere without some side effects. We've had some, but really very manageable. We had no grade 3 or 4 toxicities, and most of the toxicities were at grade 1. With respect to pharmacokinetics, ARV-471 demonstrates dose proportional increases in exposure that far exceeded the levels that were efficacious in the preclinical breast cancer models, as represented by the orange lines toward the bottom of the graph, so this one. ARV-471 also has a half-life of 28 hours that supports daily dosing. So back to pictures and sort of fun stuff. This is it here on, I would say black and white, but it's in full color. You can see that there is evidence in a patient of ER degradation. The high level of drug exposure that we just showed you translated, perhaps not so surprisingly, into seeing significant ER degradation in biopsy specimens. And what you can see here is a stained biopsy sample from samples from a patient who was treated at our second dose level cohort of 60 milligrams with ARV-471. So on the left is the patient's pre-treatment biopsy, and on the right is a biopsy that was just obtained 40 days after starting ARV-471. The reagents used to evaluate the biopsy specimens stain estrogen receptor red. What jumps out, of course, is all the red. Actually, to me, it looks kind of pink or purple, but that's because it's on top of blue. But all the red or pink here is really absent after the patient has been treated. Pretty exciting stuff. When we look a little further at several patients, we were able to get 5 paired biopsies, and we could see that ARV-471 was significantly degraded, up to 90%. And you can see here the varying range. The average degradation in these 5 patients was 62%, which is actually better than the 40 to 50% degradation achieved at the highest doses of fulvestrant. What's also exciting is that in 3 of the 5 patients, estrogen receptor was brought down to the lower limit of detection, representing complete or near complete elimination of ER in the tumor. As a final point, ER degradation was seen regardless of whether ER was mutated or not, consistent with our preclinical data. We were really excited about these findings to show that ARV-471 really does degrade estrogen receptor. And that was only from our first 3 dose levels. Most importantly, this ER degradation effect has really already translated into treatment benefit. And I'm going to show you that in a minute. So once again, we were, amazed to see and excited to see a confirmed partial response in a patient who received 471 at a dose of 120 milligrams. And this patient had been already treated with most of the standard treatments available for ER-positive breast cancer, including 2 different investigational therapies. The patient that were targeted for ER. The patient's tumor was found to have a mutation in the ESR1 gene. On the left, you can see a summary of the various treatments. Next over, we see that on the left, what I'm showing you is the left, there's a CT scan, which shows a large mass. This is the patient's liver. And you can see that there's a large sort of, less color, if you will, and that represents a mass that's in the front of her liver. And then there's another, what we call lesion right here towards the back. After 4 cycles of ARV-471, the patient had a substantial reduction of both lesions. This response again was remarkable given everything I've told you about the low expectation for responses in patients who have progressed through prior CDK4/6 inhibitor therapy. In fact, in a study of SOLAR-1, which looked at fulvestrant after CDK4/6 therapy, there was only a 20% response. So we know that it's unlikely to, we're unlikely to see much from an ER targeted therapy. Excuse me, stepping back, to look at the response data across all the patients with at least one baseline measurable tumor and at least one on-treatment scan, we see clear evidence that 471 has activity. You can see that as shown over here. So up here is tumor growing, and going in this direction is tumor shrinking. In this waterfall plot, each bar represents a patient, and the color indicates the dose level. Bars, as I just mentioned, the bars that fall below the 0%, which is up here, indicate the tumor had reduction, and that was shown by either CT or MRI. And the prior therapies are listed below. In addition to the one confirmed partial response, there are 3 more patients with a 30% reduction in the tumor size, which is the cutoff for a RECIST response. Of the 3, 2 patients had unconfirmed partial responses and provide further evidence of the efficacy achieved with ARV-471. In late-line cancer settings, doctors and patients measure not only tumor response or shrinkage, but rather the total benefit a patient may have from therapy. And that includes really staying stable. This is a composite indicator called the clinical benefit rate, and it's comprised of complete response, partial response, and stable disease that has persisted for more than 24 weeks or more. For ARV-471, we see a clinical benefit response rate of 42%, and you can see that here in this swimmer's plot. And we really can possibly compare that to, again, what I just mentioned to you about a 20% rate of clinical benefit seen with fulvestrant in a similar population. Notably, 3 of the 5 patients who experienced clinical benefit had previously progressed on fulvestrant, and a 4th patient progressed while receiving an investigational SERD. Because CVR takes into account the duration, I just want to mention the numbers on the slide. Because CVR takes into account the duration of tumor control at 24 weeks, this analysis at the time that it was done really included the patients who had had an opportunity to hit that 24-week mark. So that's why it's fewer patients than what I was showing you in the safety data prior. Based on these data, with activity seen at even 60 milligrams daily, we've initiated a phase 2 trial, ProTAC, with a once oral daily dose of 200 milligrams. To put these into context in terms of where we see, 471 and its activity, I just want to round out and show you that in December, we really tried to do an apples to apples comparison to the clinical SERDs. This is, again, this class of investigational agents that are selective estrogen receptor degraders. And here we're comparing to their phase 1 data as well. Going from left to right, I think there are 3 main points. Going from left to right, we see that 471 enrolled the most resistant patients, as with our help from the FDA, in that they had all received a CDK4/6 inhibitor. And still, we have one of the highest CBR rates that are shown in this group. Secondly, our safety profile, again, as I alluded to before, has just been excellent. One thing that's different about breast cancer therapy compared to some other cancers is that patients on hormonal therapy will stay on them for years. In the upfront setting, it can be 5 years, and in the earlier setting, it can be even up to 10 years. And lastly, as the only ProTAC in development for ER-positive breast cancer, we're really thrilled to have data showing that ARV-471 accomplishes what it was designed to do, and that it actually degrades ER. The fact that it does it more than we would have expected, more than fulvestrant, which is what we predicted, is also really exciting. To sum up, ARV-471 has a profile that really puts it on a path towards being best in class. And while that's a term that's used in pharma, what I can say is, if it's great and it's safe, it's just an incredible contribution to patients. And so we're very, very excited to move this forward as quickly as we can and into patients. And of course, we all know that nothing can be done without, frankly, too many people to even count. But I really want to acknowledge and thank all of my colleagues at Arvinas including Larry Snyder, who's joined me today, for just the incredible hard work, perseverance, innovation in discovering and bringing to the clinic this completely novel therapeutic modality that you've all been working on. With that, I'd like to thank you for your attention, and I'll take any questions that you might have.